Hi guys, these are the rest of notes 3 on passive transport, cell membranes, all of that good stuff. Just a reminder, on Friday, because it's been a little while, Friday we were talking about how cell membranes can be a fluid mosaic, meaning that things like proteins and carbohydrates, they can move and go all over the place. They don't have to just stay right here. They could end up back here or end up way over here, but they are allowed to move wherever they need to go because the cell membrane is really what controls what comes into the cell, what leaves the cell, what gives protection to the cell, and what gives a lot of support to the cells. So they can move wherever they need to go, these proteins and carbohydrates, because they will be moving to where they're needed, what they're supposed to be doing for the cell. And then we talked about how passive transport is when we have things moving through our phospholipid bilayer without any energy, mostly because those molecules are usually pretty small. And they always move down the concentration gradient, meaning that we're going to go from an area where there's a lot to a little bit, so down. And it could be down going the other direction. Down doesn't mean top to bottom, it just means from high to low until we reach equilibrium where we have about the same amount on both sides. So one type of passive transport is simple diffusion, where we talked about how these are really small molecules or they're hydrocarbons, so they're not going to get stuck in these lipid tails that are hydrophobic and don't like water. And how if we have stuff outside that needs to move in, then we say our gradient is moving into. And then if we have a lot inside and it needs to move out to reach equilibrium, then the gradient is moving out up. So things are moving out of that direction. And then we talked about another type of simple or passive transport called facilitated diffusion, and this is where we're going to need the help of a protein channel, kind of like a tunnel that's designed to allow a specific type of molecule to pass through that protein to go from one side or the other to the other within the cell membrane. That's what allows the membrane to be called selectively permeable, because they select for certain molecules sometimes to come through. Sometimes they just easily move through, but sometimes we have to select which ones are able to come through based on their size. And then a type of facilitated diffusion we talked about was osmosis. And osmosis was where maybe we can't let bigger molecules like sugar molecules go across the membrane, so water will be the thing that diffuses across the membrane, not the particles, but the water can also diffuse across the membrane to help make this equilibrium of the water to soluble or solute, be more in ratio with the other side of it on the other side of the membrane there. So that's where we left off on Friday. We're going to pick up there and talk about tonicity. Tonicity it's kind of a silly kind of a word, but it's a word that we're going to use quite a bit now for the notes, and then also when we go back to our lab. Tonicity is the ability of a solution to cause a cell to either gain or lose water. And it really kind of depends on the inability of the solute to move across the semi-permeable membrane. So in this case here for the beaker with the sugar molecules, the solute wasn't able to move across the membrane, so the water has to move across the membrane. And that's going to affect our tonicity, our ability to gain or lose water. So because the solute doesn't move, we're going to lose water from this side to the other side, and then there's where tonicity plays a role. So this was our inability to keep the water here because it needs to move across the membrane to balance out that ratio of solute sugar molecule to water ratio. Water will move in and out depending on the concentration of non-penetrating solutes. So because these sugar molecules could not move, the water is able to go back and forth through that membrane, just depending on where it needs to go to make sure that that ratio is balanced or in equilibrium. We can say that the net flow of water is always in the direction towards the highest concentration of the solute. These are the molecules. like sugar in the example above. So because we need to move the water to where there's a high concentration of sugar, the water will move in that direction. Now we have different types of tonicity that we talk about, and we use different terms depending on whether or not it's a plant cell or an animal cell. So we're gonna work across the top here labeling these guys. These are all examples of animal cells and different tonicities. This one here, if the water has to move into the cell, think of a water balloon. When you have a water balloon and you fill it up, you fill it up, you fill it up, and eventually it's gonna burst open. When we have a ton of water moving in, causing it to burst open, this means that it has lysed. And we've talked about lyse, that prefix before, like hydrolysis, when we talked about splitting apart macromolecules using water to burst it open. We've talked about lysosomes. Lysosomes cause things to break apart and burst open. So that's what we're talking about here. And this is when our concentration is hypotonic. Hypotonic means that water, the concentration of the stuff inside the cell is a lot higher than outside the cell. So the water has to rush in to try to balance that out and reach equilibrium. But so much water rushes in that it bursts open really fast and pops open. So that's hypotonic for an animal cell. That term is called lysed when it bursts open. If we have a solution outside of the cell that's a lot higher than inside of the cell, causing the water to move out of the cell. So the water inside the cell recognizes, hey, there's a lot of big molecules outside. We need to rush out so we can make equilibrium out there and make sure everything's in balance. Well, when all that water rushes out, then our animal cells, the term for that shape is, or that action, nothing really crazy here. It's just called shriveled, because it looks like it's kind of like sunk in on itself, but really what's happened is all that water rushes out and then, yeah, it kind of shrinks in and shrivels up. Kind of like a raisin. If you think about a raisin and taking all the water out of a grape and then you end up with a raisin, that's this one here. And that's when our solute out here, the solution out here, is hypertonic, meaning that there's too much of the solute particles outside of the cell, so the water has to rush out. Now, if we're in balance and everything is pretty much at equilibrium inside the cell and outside the cell, and we don't need to have water rushing out or rushing in, they're just kind of at a balance of equal movement in and out, then that's normal. And that's what we call the cell. And this is when we are isotonic. Isotonic means that the solution concentration, solute concentration inside and out of the cell are the same, iso. So animal cells in a hypotonic solution will lyse or burst. In an isotonic solution, they're just normal. In a hypertonic solution where the water rushes out, then they're shriveled. For a plant cell, it's a little different. Because remember, plant cells have those cell walls on the outside of their cells, and then they have the cell membrane. So that cell wall is going to give them extra support because they need to be a little bit more rigid than an animal cell, and that plays a really big role in what happens here. So in a hypotonic solution, where there's more stuff inside the cell and the water has to rush in, it's still going to get bigger, 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 but because we have that large central vacuole, it can hold a lot of water. And it actually makes it normal for a plant cell for the water to rush in. And we call that turgid. When the water is rushing in and there's more water coming in than there is outside of the cell at that point. And that's because the plant cells really need that water. It provides turgor pressure. I've mentioned that before. But the water that's rushing in is all going to push out on these cell walls all over the place and making it really rigid and tough so it can survive growing in different kinds of elements. And we get nice strong plants from that. That does not mean overwater your plants. It just means that plants do like that water. They can keep that water in that large central vacuole, and they can help provide a lot more support. Now, if we go the way opposite end of the spectrum over here, and we have more of the solute particles outside of the cells, and the water has to rush out, this is something that's called plasmolyzed. And plasmolyzed means that the water rushes out. It doesn't shrink in like we have for our animal cells, but instead what happens is the cell membrane, which is this part right here, the cell membrane will actually pull away from the cell wall, and it doesn't detach, it just pulls away so that the whole cell is a lot more smaller inside. It is like it's shriveled, but because we have two layers of protection and support, the cell wall and the cell membrane, the membrane just pulls away from the cell wall, and it'll keep doing that for that situation. So again, that's in a hypertonic solution where there's more of those solute particles outside, so we have to have the water inside the cell rush out to try to balance it and reach equilibrium. And then in an isotonic solution where we have a normal amount of water, regular equal amount of water I should say, moving in and out equally, the solute particles are about the same on both inside and outside the cell. This is just what we call flaccid, because it's just their regular sort of cell, not as full of water in the central vacuole as it would be for turgid or a normal cell, but there's some in there, and it could use more water, but it just doesn't have more water quite yet. Okay, we're gonna flip over. We've got one more page, and then I think we're just about done. So just to review, passive transport is where particles can just simply diffuse across. We can use a protein channel through facilitated diffusion, allowing only specific shapes to come through. Or we can have active transport, which we're gonna get to later. So this is where energy is used. We're not gonna talk about this yet. This is a completely separate part of notes, but just to show you guys, we have to have energy to allow things to come in. For right now, we're working on simple diffusion and facilitated diffusion. Simple diffusion and facilitated diffusion as examples of passive transport. And another example of facilitated diffusion is osmosis, because water would need to come through aquaporin if we're talking about large amounts. All right, so now another example here, just so that you guys can see how this works. The dots inside the diagram are going to represent solutes. So maybe there's sugar molecules or just bigger molecules that are moving in and out of the cells. The diagram shows cells placed in hypotonic, isotonic, and hypertonic solutions. So the solutions are out here, respectively. So if we put this cell, or out here, I guess I should say, around the round part, the cell part. So if we put this cell in a hypotonic solution, the water is going to move in to the cell because there's more of that solute in the cell than outside. So the water has to rush in to try to balance out the ratio of water particles, molecules, to these solute particles inside here. In an isotonic solution, we have an equal amount of water to solute ratio. So we're gonna say that we can have H2O in here going in, but we can also have H2O, water inside the cell, coming out. And I know that's really small, but just label H2O and H2O. So in and out equally. And then in a hypertonic solution, we have more of our solute outside of the cell. So our water inside of the cell, H2O, is going to need to rush out to try to reach equilibrium over there. So within a hypotonic solution, that means inside the cell there's more stuff, so the water outside the cell has to flow in to make it reach equilibrium. The solutes outside are less than the amount of solutes inside. Water moves into the cell. It will swell and burst. In an animal cell, you could call it cytolysis. We just wrote lysed for on the other side of our notes, but, or cytolysis is what it's called, cytolysis, lyse. So cytolysis is the word, but the root part of this I really want you to just know is lyse. Lyse means to burst. In plant cells, if this were a plant cell, then it increases our turgor pressure. The cell walls do not burst, they're just going to get turgid. In an isotonic solution, if we put the cell in there in an isotonic solution, then we have equal amounts of solute particles inside and outside. Water is equally moving both in and out of the cell at all times. There's really no change in the size. A really good example of this are our red blood cells in animals, or humans really, because our blood cells need to have that shape to be able to flow more freely through our veins and our arteries, and we'll talk about the shape of cells and what happens when we don't have that shape and we're not in an isotonic solution later on down the road. And then hypertonic solution, again, all of the stuff, the solute particles outside of the cell are going to be in greater quantity than inside. So that means that the water inside the cell is going to want to rush out to try to balance this stuff out and make it reach equilibrium. In an animal cell, when it leaves, that process is called crenation. But remember, we said that it's just a shriveled shape. So you could just say that it's shriveled. And then with plant cells, when the water rushes out, the cell membrane is going to pull away from the cell wall. That's also called plasmolysis. You could say that it's flaccid there, but because it's moved away from the cell walls too. A lot of times you'll see that term used interchangeably even when it's in an isotonic solution, just because it's not as full with the water and reaching that turgid pressure as it is here in a hypotonic solution. Okay, that's it for our notes. We're all done, nothing on the back. So what you're going to do next is use those notes to help you finish the rest of your lab questions from Monday's lab, answer all the whys for day 2 and anything else that had a star, and then you're turning in your lab today by the end of the period. And keep these notes out because you're going to need them for the next assignment once you've turned in your lab. Thanks.